Hello, grade 11. We're gonna proceed with our lesson that is the cell cycle. As we said previously, when we talk about the cell cycle, we talk about two phases, interphase and mitosis. Interphase is the phase where the cell is preparing itself to divide. Mitosis is the cell division. Now, as we also said previously, when we talk about interphase, we talk about three stages, G1, S, and G2. I said before that during G1, the genetic information or the DNA is in the form of chromatin. In the S phase, the DNA or the genetic information doubles, okay? It doubles, it duplicates, or we say replication takes place. Now, in the growth phase 2, that is the G2, we have chromosome made up of two chromatids. So, if I talk about quantity of DNA or mass of DNA, I can say that during growth phase 1, just a second, I'm picking my pencil. Okay. I can say that during G1 phase, growth phase 1, the quantity of DNA inside the nucleus is Q. Okay, it is Q. During the S phase, the quantity goes from Q to 2Q, which means that it is duplicating, it is replicating, it's being doubled. Why it's being doubled? For the cell to divide, it's preparing itself to divide. So I have in the growth phase 2, G2, I have officially chromosome made up of two chromatids, which means that the quantity of DNA here is doubled, okay, before the cell divides. Now, just a second. Okay, as you can see here, in the prophase, for example, this is 2n equal 2, okay? This is 2n equal 2, which means that a cell with two chromosomes. In the prophase, we reach 2q, which means that the cell prepared itself during interphase. In G1 it was a Q, in the S it was Q going to 2Q, being doubled, replicating. In the G2 phase we have 2Q. So the cell is ready to divide. We enter the prophase, the first phase of mitosis, with 2Q, double the quantity of DNA. In the metaphase, it is also 2Q because chromosomes are lining up along the equatorial plate. The quantity of DNA remains the same, okay? Now, during anaphase, as you know, what happens to the chromosomes? The chromatids of each chromosomes are separated, as you can see here. Each chromatid moves to opposite side of the cell. So, I can say that the quantity of DNA is changing from 2Q to Q. It's being reduced. Why it's being reduced during the anaphase? Because it's preparing itself for telophase. It's preparing itself for telophase because during telophase we have two daughter cells, okay? Each cell having the same number of chromosomes as the mother cell, but in the diploid state, as you can see here. In the diploid state, in the haploid state, sorry, not in the diploid state. In each chromosome of one chromatid, not of two chromatids. Okay? Then the quantity of DNA was reduced from 2Q to Q for each daughter cell in the telophase. Okay? Logic. Here in the prophase, you see each chromosome made up of two chromatids, while in the telophase, we have two chromosomes, right? But each chromosome is made up of one chromatid, not of two chromatids. So the quantity of DNA was reduced to half, from 2Q to Q. Okay? Now, this is what I said, variation of mass or quantity of DNA during cell cycle. Consider here we have, for example, 2n equal 4. Just a second, I'm picking my pencil again. So, here it is. Okay. So, consider we having cell with 4 chromosomes. As you know, here G1, as the cell is replicating. G2, we have chromosome made up of 2 chromatids. During prophase, we have 4 chromosomes. Here they are. Metaphase, these 4 chromosomes are lining up along the equatorial plate. During anaphase, what happens? What happens to the chromosomes? Okay, they are 4, but here the drawing is not clear. Each chromosome divides, the centromere of the chromosome divides, and each chromatid moves to opposite side of the cell during anaphase. We call this polar ascension. Then we have the telophase, where we have 2 daughter cells, and each daughter cell having the same quantity of DNA as the mother cell, but each chromosome is made up of 1 chromatid, not of 2. So it is haploid, not diploid, okay? Okay. Here is a graph that shows the stages of cell division. The first thing that attracts your eyes is that if you look here, you can see that this is G1, S, and G2, okay? So, till here, we're talking of interphase. So, can you see that this is the time in hours? Can you see how long the interphase take? From 0 to 18 hours. Like 18 hours is the interphase, while the mitosis is like 2 hours. So, the longest phase in the cell division is the interphase, is the phase that is the cell preparing itself to divide. Just like you girls, when you're preparing yourself to go out, the preparation takes time more than the outing. Okay, so look here, guys. Okay, so here is the G1 phase. The quantity of DNA is Q. It didn't double yet. This quantity is being doubled in the S phase. It's going from Q to 2Q until we reach the G2 phase, where we have double quantity of DNA, which means that the cell is ready to divide. Huh? You should memorize this. You should know how to schematize this graph, okay? So, G1, S, G2. Directly after G2 comes the mitosis, comes the cell division. So directly after G2, here we have prophase metaphase. Why here prophase metaphase? Because here the quantity of DNA is still doubled. When the quantity of DNA starts reduction, okay, it goes from 2Q to Q. It is being reduced to half. So this is the anaphase. Now, at the end of anaphase, the quantity of DNA is Q. So here begins the telophase. 2 daughter cells, same number of chromosomes, but in the haploid state. Okay? So this was a graph showing the variation in the quantity of DNA as time and hours. Okay. Now, let's talk a little bit about DNA. What is DNA? What does this term stand for? DNA is deoxyribonucleic acid. The name is deoxyribonucleic acid. When talking about the structure of DNA, this you should memorize it, huh guys? DNA is a double-stranded DNA. Double strand. Made up of two-stranded molecules, okay? So DNA is double-stranded. It is hair-like structure, especially the curly hair. It is hair-like structure or molecule twisted around itself in a helical form. Let me tell you what that is. Okay. Okay. So it is twisted. You see what is twisted? This is twisted. So DNA is twisted like a helical form. It is double-stranded. You should know this information very well, okay? DNA is about 2 meters long. It is very, very, very long. But due to the condensation mechanism, it is condensed. مضبوبة. Due to the condensation mechanism, it is transformed into chromosomes to be fitted inside the nucleus. So the DNA is like 2 meters long. It's very long. But for its condensation, it is put inside the chromosomes to fit inside the nucleus, okay? This genetic information is condensed inside the chromosomes in order to fit, okay? This is the DNA. It undergoes condensation. It becomes packed, if you want. It becomes tightly packed in order to fit inside the chromosomes, okay? So DNA is the deoxyribonucleic acid. DNA is double-stranded. It has a twisted shape or helical form. It is 2 meters long, but it is condensed inside the chromosomes in order to fit, okay? Now, composition, the chemical composition of DNA, which means that what is it made up of? DNA is composed of nucleotides. You should memorize these. DNA is made up of nucleotides. Okay, so DNA is made up of nucleotides. Each nucleotide is formed of each nucleotide. So DNA is made up of nucleotides. Each nucleotide is formed of sugar called deoxyribose. So deoxyribose sugar, denoted as D. Phosphoric acid that has the phosphate group, so it is denoted as P. And nitrogenous base. So DNA is made up of nucleotides. Each nucleotide is made up of deoxyribose sugar, phosphoric acid, nitrogenous base, okay? And when we talk about the nitrogenous base, we have 4 types of nitrogenous base. The 4 types of nitrogenous base are A, T, C, G. A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. Again, I'm going to repeat. DNA is a sequence of nucleotides, is made up of nucleotides. Each nucleotide, in turn, is made up of deoxyribose sugar, phosphoric acid, and the nitrogenous base. What are the 4 types of nitrogenous base? Adenine, thymine, cytosine, and guanine, okay? If you look down at the nucleotides, the first nucleotide is A connected to D connected to P. This is A is the adenine, D is the deoxyribose sugar, and P is the phosphoric acid. So here we have adenine. And the second one, C connected to deoxyribose sugar and to phosphoric acid. So C is the cytosine. The third one is T connected to deoxyribose sugar and to phosphoric acid. T is the thymine. So here, which nitrogenous base we have? The thymine. And the last one we have guanine, since G, nucleotide G, nitrogenous base G is connected to deoxyribose sugar and to phosphoric acid. This is what I want you to know for this session. Study well, guys. Copy this on your copybook and see you tomorrow.